Okay, there we go. So, we've gone through chapters 13, 14, 15, and 19 for the electron transport chain. So it's time for our first exam. That's a lot of chapters, right? That's a lot of stuff. And it's pretty crazy because we could probably have, exams every 2 chapters in here, but then we'd have something like 7 exams for the semester. I don't think we'd have enough time for the exams. I have to put them all outside of class or something. But anyway, what do I have here? Things to know by memory. So, yeah, there are. So I think what I'm going to do is split up this exam next Tuesday. So part of it will be in class and part of it will be take home. Okay. So I'm trying to, with the study guide, to know things that you should know for the in-class portion. All right. So, and these are going to be our foundational principles. The take-home portion is going to be applying those principles in a variety of ways with some theoretical questions like, well, why? What happens if you do this? And so on and so forth. So from chapter 13, it looks like there's a lot here, but it's really kind of all the same stuff. It's stuff we've seen before, and all this is interrelated in here. How do changes in concentration or equilibrium affect delta G naught prime? And you might think about delta G naught prime equaling, negative RT, log of negative RT, oh geez, RT log concentration, whatever that equation in there that kind of relates temperature and concentration and all that good stuff. Yeah. Yes, it is. Yeah. So you can hear me stuttering over this stuff in here after my lovely morning. So the other thing is, what's the difference between our Gibbs free energy, with the dagger and the knot sign up there versus this, one without. And, we talk about standard conditions a lot. If you think of chemistry standard conditions, it's usually something like 0 degrees Celsius and, whatever in there. But here we redefine our standard conditions typically to be 37 degrees Celsius. Why? Because that's body temperature for us and, at least close for many other mammals as well too. But we have to keep in mind that within the organisms, within the cells, we are far from standard conditions. So there is no, but 1 molar would be standard conditions for concentrations. In chemistry, it might be 1 millimolar or 1 micromolar in the cells, but we're way off from that. So standard conditions are not really standard, is what I'm trying to say here. So a lot of these calculations where we come up with this energy is like, if we were at these conditions, this reaction would go this way, or this reaction would go that way. But when we have these pathways where we have reversible reactions, our non-standard conditions, we change things. So we're going to drive it to the left, or something else is going to happen. So, now we've changed conditions. So we drive it to the right. Okay. And that's why the distinction between our standard free energy versus our, actual free energy becomes important, because that tells us which way our pathways are going to run. If you look at that whole glycolysis gluconeogenesis thing, depending on what the enzyme sees the most of, it's going to treat, I'm going to treat this as a substrate and release this as a product. But if our product is really high concentration, I'm going to treat that as a substrate and convert it back into, original substrate in there. Okay. It's always a good idea to know various functional groups. I'm not going to say, is this a carbonyl group or an aldehyde group or a ketone or a sulfhydryl or something like that. But, just refreshing that organic knowledge is going to be useful in there. And like I say, I stay away from testing you on mechanisms. It's like, okay, well, the lone pair on the sulfhydryl group of this cysteine in this enzyme is going to make a nucleophilic attack on this carbon. And, I'm not going to, explicitly test that knowledge, but I think it's helpful if you can visualize that. So all those little slides where you have like the little arrows of here's where the electrons go, and here this bond flips up here and this one flips over this way, that explain that mechanism, those are useful because that helps you figure out, okay, this is why I'm taking a 6 carbon, sugar and splitting it into 2 3 carbon sugars. Okay, this is not something that's easy to do because we're breaking a carbon-carbon bond. So breaking and making carbon-carbon bonds are kind of like the hard things that the body does a lot of. So we have to charge them with phosphate, which is very electron withdrawing. So we end up making a carbocation. So we can have this carbanion somewhere, and so on. So, but this all ties in. Knowing these functional groups helps you explain how we make energy available, because we have these high energy compounds that when they react will release, they're very exergonic, they'll release a lot of energy, but that doesn't just, not like we have little explosions in our cells. That released energy is used to drive an unfavorable reaction. So we go all the way back to chapter 1 in Biochem 1 and say we are going to couple favorable reactions with unfavorable reactions to drive them forward. Okay. It's kind of like dragging your friend to class. I don't know if you ever had a roommate who would like to sleep in. And come on, it's time for class. Get up, let's go. And, I probably could have used one of those roommates when I was. That's funny because my freshman roommate and I probably sort of took turns trying to motivate each other to go to class, but we skipped way too many classes as freshmen. Yeah, it's hard being a freshman. So. High energy phosphate compounds. So you've got, of course, ATP, all of your nucleotide triphosphates, goes out hydrolysis of inorganic phosphate or pyrophosphate. Cleaving those phosphate bonds just releases a lot of energy. You see the same thing with phosphoenolpyruvate in creatine as well. So those are some high energy compounds. Basically, when you break those bonds, you have lots of other energy in there. Apparently, I just turned 14 again. We break those bonds. Oh, yeah. My voice crack on the recording here. Post that to the tickety talk. Okay. Yeah. It's been done. It's been done. I got to turn myself into like a can of Mountain Dew or something there. Come up with a new fill. Oh, I get a sponsorship. That'll be it. I'll be sponsored by this lecture is sponsored by Mountain Dew. I used to joke about that because there was one other class where the example they had, they used Sprite, and like there were like 3 different slides that had cans of Sprite on there too. And it's like, clearly this lecture is sponsored by Sprite. So, all right. And I'll see if I can do that for money here. Let's see if like Coca-Cola company I'll put your logo on all my slides. No conflict of interest there. Okay. So again, our coupling of reactions, exergonic, inorganic processes are coupled in there. You maybe I'll give you like a short pathway, or I'll make up a pathway and provide delta G values. And I'll say, which way is this pathway going to run? Or maybe I'll say something like, if you look at like 3 or 4 reactions in a pathway, does one of these look like it's a good point for regulation? So maybe if it's got a massive delta G value, you'd say, yeah, that's only going to be favorable going one direction. So if it's negative, it's going to go forward. If it's big and positive, it's only going to go the opposite way. So that would be something that would probably need some help, being driven against its gradient, or it may be effectively irreversible. So that would be a good enzyme target for regulation. Okay. And I like the analogy. We can go back to all sorts of the analogy. How do you control an octopus with 8 arms attacking and all that? You control the head. Same way in military structures, you take care of the head. Take the leadership in here. So in our metabolic pathways, if you identify the key regulatory enzymes, you control those enzymes. You control the whole pathway. So that's what we're getting at here. Okay. So the last thing out of chapter 13, it also ties directly in 19, is why is NADH important? NAD plus, NADH, and similarly NADPH and NADP plus and FAD and FADP. Those are our electron transferring compounds in there. So if they're transferring electrons, accepting and transferring electrons, they are driving processes in there. And I always think back to my basic undergrad organic, but I had a year of grad school in chemistry. I took an advanced organic chemistry class, which was fun in the sense that I don't really remember a whole lot about it, but I remember the key was follow the electrons. So just like if you want to figure out who controls something politically, follow the money, follow the electrons. If you can figure out where the electrons are going, you can predict what products are going to be made. So the same way our electron compounds here, NADH and FAD and NADPH, are going to be the ones that are shuttling electrons where they need to go. So they're going to be the ones that are providing the energy for a lot of these reactions. So by shuffling those electrons through the electron transport chain, that provides the energy to pump protons from the matrix to the intermembrane space so that they can flow back through ATPase and generate ATP. So, I kind of joke a little bit that chapter 13 is like our review of first semester biochem, because it all comes down to coupling, all right, in there. Jumping ahead to chapter 19, the electron transport chain, I want you to know, the general structural arrangement of mitochondria. It's got that dual membrane. There's a mitochondrial membrane. It's like, oh, yay, big deal. The inner membrane, the intermembrane space and the inner mitochondrial membrane are kind of where it's at, because that inner membrane is what separates the high proton concentration or low pH intermembrane space from the matrix. You're just basically sucking up protons from the matrix, putting them in the intermembrane space so they can flow back in there. Okay? Being able to describe the first four complexes is important. I always joking, complex 1 looks like Florida. Okay, that's not a good description, but it's got the proton pumping section in like the panhandle, and then the other little section down here is where the electron transfer takes place, where it has the substrate locations for NADH and various cytochromes that are in there. I'm not going to ask you guys to tell me, okay, well, this is cytochrome A in here, and you got cytochrome A and cytochrome A1, and you've got two iron, 2 sulfur complexes in one iron, one. No, that's too much detail. That's more than you need. But I just want you to know that there's a network of basically electron sinks in there, so that you can pass these electrons that come from NADH through these locations of varying electron density / affinity, so that basically electric potential. So you're basically trying to get a little charge to flow through there. So electrons are going to go from negative to slightly less negative to slightly less negative to slightly less negative, so that it flows through there. You're getting this current flow, and that's what pumps the protons out. Okay. You're absolutely going to get a question about chemiosmotic theory. This electrochemical gradient, is just massively important. It's why we live. So just being able to explain, we use the energy that comes from complexes 1 through 4 to pump out protons, so that complex 5 can let them back in. Being able to describe complex 5 ATP synthase, how it works. And under normal circumstances, if you put ATP synthase in a cup of water with some ATP and some protons, it's going to hydrolyze the ATP and make ADP and free protons and stuff like that. So that's why we build up this gradient. We're essentially making it run backwards to make ATP. Okay. All right. But really, chapter 19 I like because it's very straightforward. It goes into too much detail in Lehninger, but there's really only 4 or 5 things to take away from there. Okay. I need to call them again and again, because I've let them know, but I have not heard anything from them. So again, that's why there are that's why there are currently, as a workaround, PDFs specifically for the purpose of education available to students for this semester. Yeah. And that sucks. I know that's not the best way to do it, but I'll keep contacting the bookstore until somebody calls me back. Yep. But the other thing is that Lippincott's illustrated review is, for this semester, fantastically helpful. So I think Lehninger does a good job and a reasonably readable explanation of a lot of what we're talking about here. But as we keep going, Lehninger gets denser and denser and denser. And Lippincott's maintains readability. So when we get into like the, like, I just don't care about fatty oxide synthesis at this point, Lippincott will be your friend. It will get you through there. Lehninger is a good textbook. Lippincott as an illustrated review is what all the first year med students use to understand enough to get through their med school biochem. It's probably, in my opinion, and I've read 7 or 8 biochem review books, some of them like only 100 pages. Lippincott's is 500 pages, but it is a phenomenal review. It will get you about 80% of what you need to know, but it's also really readable, so that when if you get stuck in Lehninger, Lippincott's is your friend. Plus, I've been known to pull questions from Lippincott's for some of the questions that I use on exams. There's one I got to find. I got to check and make sure, because now there's so many editions of Lippincott's. Is like every year or every other year there's a new edition of the review out there. And there's one question I like to ask, and I was like, well, crap, I don't even know if they use that example anymore. But so I got to check on that. All right. So those are the first 2 chapters. Background overview of bioenergetics and the electron transport chain. So far, not too bad. Chapter 14 gets a little hairier, but a big chunk of those slides that we have in there are going over the specific mechanisms of the reactions in excruciating detail. So what I want you to really take away from this, what I want you to have at your fingertips, is talking about glycolysis. But what happens to glucose when it gets into the cell? So first thing we got to do is phosphorylate it so it can't get back out. All right. Trying to think of something. And I do not have a good analogy for that. I have bad analogies for that. Yeah. So yeah, my favorite one is like the crazy cat lady who put shock collars on the cats, so once they're in the house they can't get out. Yeah. Pleasant one. All right. But then what happens to glucose? And keep in mind there are like 4 different things that I think we talked about that can happen to glucose. We can store it as glycogen. We can send it through the glycolytic pathway to make energy. It can go down the pentose phosphate pathway to make nucleotides, and we can use it to make proteins as well too. And I think that might be it. Yeah. Okay. So I do want you to know the 10 steps of glycolysis, but really the 3 enzymes are the big ones: the hexokinase, the phosphofructokinase 1, and the pyruvate kinase. Those are the big 3. It's maybe good to know all the enzymes in there, but they're not necessary. The 3 big ones in there. Know there are 10 steps, and know that the first 5 steps, you're putting energy into the system. You got to spend ATP to phosphorylate these components. But then the last 5 steps, you start getting energy out. You make NADHs, which can make more ATPs in the mitochondria, and you get a couple ATPs out of there from like the phosphoenolpyruvate. And of course, it also helps that you've split your glucose into 2 3-carbon molecules, so you have 2 PEPs. So that's why you get 2 ATPs out of there to make up for 2 that you spent, and then the other stuff you get another 2 out. So, all right, and then once we have pyruvate, we sort of talked about this in 14, and we talked about a bit more in 15. What happens to pyruvate? We can convert it to lactic acid. We can convert it to ethanol in bacterial cells or yeast cells. It can go in. It can feed into the citric acid cycle. It can go into making amino acids as well too. So just be aware of what you can do with pyruvate. I think one of the things we mentioned is like glucose. Glucose can make everything except fat. You can't turn sugar to fat, or can you? You get acetyl-CoA. Pyruvate, I think you make into acetyl-CoA, which can go into fatty acid synthesis in there. Again, we'll see all this coming up later on. But I may give you a multiple choice question or something where there's, which of the following is not one of the fates of pyruvate. So knowing those is good. All right. So we tend to be very liver-centric in this. What would that be? Hepatocentric? Yeah. In class. because most of the stuff we talk about is in the liver. Gluconeogenesis, we're going to see happen, pretty much exclusively in the liver. So where does that glucose come from? Well, we can take our pyruvate. So if we work out and we burn some glucose in the muscles, we make lactic acid, which gets converted to pyruvate, which gets sent to the liver, which then can go through gluconeogenesis to make new glucose, which can get sent back to the muscle, so you have more energy to keep going. I think that's why I overdid it. I spent, not yesterday because it was raining, but the day before, I was like, yeah, I'm going to split firewood just in case, it's culinary. I had some nice wood that split easily, but still a whole lot of splitting wood. And I'm like, oh, yeah. Haven't used these muscles in a while. Vitamin I, perfect. Yeah. And come in and drink a Mountain Dew so I have more energy. But I have to give my son here to come out and help, and he stacked the wood very, very nicely. So, yeah, yes, yeah. Even if he did then later on lie to his mother about what homework assignments were due, and that's why she was in a bad mood. So, and that's like contagion, it spreads throughout the house, right? I'm just another happy aspect to my brain. Okay. So, where does the glucose come from? Why is it necessary? Where does it go? And why is glucose necessary? Yeah, and specifically, there are some organs that can only use glucose for energy, like the brain, and the red blood cells. Yeah, so we got to have our sugar in there. Always makes me think of a Kids in the Hall, very old sketch comedy, hilarious, offbeat, but they had a movie called Brain Candy, which is a little weird. It's probably not very mainstream accessible, but brain needs sugar. Sugar's in candy, brain candy. I always keep that association in there. So glucose is what fuels the brain. Okay. And then you'll probably get a brief question about the pentose phosphate pathway. So I believe these are the correct figure numbers, but I may need to update these. I definitely will for next year when I actually get the current textbook in there. But figure 1427, I believe, describes the pentose phosphate pathway. And it's really just the idea of, hey, we're going to make these pentose sugars, which are going to be the skeletons for our nucleotides in there, right? And of course, that's where we get NADPH. So, and then finally, we get to our last chapter in there, chapter 15, which is where it kind of starts getting crazy, right? Because now we start talking about regulation, and regulation is what we're going to talk about for the next 2 months. It's like a finance class. We've got regulations, right? Regulation is absolutely critical to living organisms. We got to make sure that we are responding to the various changes within the organism, within the environment, within the availability of food sources. You think about different bacteria that, it can grow on glucose. It can grow on starch. Well, it's kind of the same thing. It can grow on lactose. It can grow in an aerobic environment or an anaerobic environment. You think about different organisms that can that have the choice to reproduce either sexually or asexually. How do they make that decision? A lot of that depends on the environmental factors, what's available, what they can do, and the whole goal is to keep the organism alive so that it can reproduce. So all this goes back to regulation, which is why we are going to talk about the regulation of these different pathways. And we kind of looked here, how do we determine whether we're going to go through glycolysis or gluconeogenesis? Well, if we have lots of glucose in here, chug my Mountain Dew, woo, got glucose in my veins, I'm full of energy. Caffeine helps too. But that's going to trigger a response in there saying, hey, we got lots of glucose. We're going to motivate transport molecules to the surface to bring that glucose in. We're going to release hexokinases from their sequestered section to phosphorylate them so that they stay in. If we have already a high energy state, we're going to store these for later. But if we need energy, if I've been out chopping firewood, straining muscles, then it's going to say he needs more energy. He's got to cut more wood. Then we're going to shunt that into glycolysis so we can make ATP, so that we'll have energy in there. Along with the availability, and conversely, if I wake up in the morning, decide I'm gonna skip breakfast. I'm gonna go out and chop some more wood. I'm gonna not have a lot of circulating glucose in there. So we're gonna see a gluconeogenesis happening, especially as I'm working out. I'm making lactic acid in my muscles. That's going to go to the liver, and we're going to upregulate gluconeogenesis. And both of these pathways are antithetically regulated by the same mechanism. So we'll see phosphorylation. Phosphorylation turns on one pathway, it turns off the other pathway. We saw that when we talked about the comparison of phosphofructokinase 1 with the fructose 1,6-bisphosphatase. There's essentially a bifunctional enzyme in there. When it's phosphorylated, it catalyzes the one reaction going one direction. When it's dephosphorylated, it catalyzes the opposite reaction in there. Okay. And I'm going to use words like that quite a bit, like bifunctional enzyme. It basically, it can bind a substrate and convert it, substrate 1 to product 1, or you can bind substrate 2 to product 2. So it can do a couple different things. We have a trifunctional enzyme coming up as well too. Yeah, we'll get crazy. We're gonna get crazy in here with our multifunctional, weirdly efficient, purposefully inefficient for higher regulatory control, but very efficient in that we only make one product. I like to say I compare it to organic chemistry labs all the time. It's like, I made 15% of what I wanted. Yay, that's really good. It's like here it's 100% or bad things happen. Other things come in and take care of whatever is not correct. We also have to keep in mind that our relative concentrations of both ATP and AMP will regulate a lot of these molecules as well too. Many of the molecules, particularly in energy production or consumption pathways, will have allosteric binding sites for ATP or ADP or AMP, and depending on what binds there, will either ramp them up or slow them down. If we compare the ratio of ATP to AMP, that is a good indicator of the overall health of the organism. So if we have a very high concentration of ATP relative to AMP, we are in an energy positive state. We're good. We got all the energy we need. Got what I need. I can work on storing energy now. The other college students, you probably live off campus and do your own cooking, right? Yeah. So, when you're single, you can make like a pot of chili and just have chili every day for the week, right? And it's one day of prep work, and I got to put in energy to make this. I'm gonna make something that's good and tasty, and it's gonna stay good for several days. So you do the same thing with pasta, just heat it up in there, something like that. So you kind of prep. I actually had friends; these are mainly engineers who would like make a week's worth of sandwiches. They would spend their Sunday and make, here's all my, yeah. I mean, it was, yeah, that's what I would think too. It's like, okay, that's a little too much, it's like, I've been called extra, but I'm not compared to some of the people I grew up with. I'm like, okay, I'm a step above, but I have people who are downright obsessive. But I mean, most of these people are engineers too, so that is a stereotype that seems to hold some water within engineers. You think chemistry majors are weird, or physics majors are weird. Engineers are a whole new level. Yep. It bothers me because in my house there is a set of switches. Like I keep thinking, I'm just gonna flip these around at some point, fix them. But all the light switches have to be down for the lights to be off, except in the one hallway where those two light switches both have to be up because the others will be down then. But I can't have one up and one down. That's just wrong. Yeah. And I notice all this crap. There's to go back to our ATP AMP levels, the big thing is that AMP is probably a better sensor because physiologically AMP is at about 2% of the overall concentration of ATP in a typical mammalian cell. So if you change a little bit of ATP into AMP, you have this little teeny tiny change in ATP overall concentration, but that might double or triple or 6 times as much AMP suddenly shows up there. So AMP is kind of like your first indicator. As those AMP levels increase, the body starts saying, "Whoa, we're using a lot of energy. We got a lot of AMP here. We may need to make some more." So that's kind of our initial sensor. But this overall ratio helps as well too. And you can see if AMP levels are very, very low in the cell, a small change in those, you're going to see this big change in that ratio corresponding to that. All right. I think figure 15.7 is the isozymes of hexokinase, or maybe actually, no, sorry. It's the hexokinase, phosphofructokinase, and that phosphohexoinositol. One of the enzymes in the pathway that is not regulated. Okay. So I like this because it shows how differences in substrate concentration, in this case glucose, can affect the levels of the enzyme. So I chug my Mountain Dew, I'm going to see levels of hexokinase in my cells shoot up like mad. So like, oh, we got lots of glucose, we got to phosphorylate it all. PFK1, our first committed step, our gatekeeper to glycolysis, is also going to ramp up. It's like more glucose, we're going to use some of that for energy. So we're going to respond to that. And then that phosphohexoisomerase, I believe, is what that one is. That's the one that just basically interconverts it from maybe fructose 1,6-phosphate to fructose 6, glucose 1, glucose 6. I can't remember that, but it just has no effect. It's like, oh, more glucose. Okay, job security. So it's just going to keep going working. It's got a basal activity, right? And if there's lots of glucose, it's going to be full more often. It's going to be saturated and just be converting in there. If there's not a lot of glucose, it'll go the other way because it's one of the reversible ones you might see in gluconeogenesis. So it's not responsive at all to the substrate. If we look at the regulation of glycolysis, again, looking at these regulation points, enzymes 1, 3, and 10 for glycolysis. And then if you go backwards, 10/9, you got pyruvate carboxylase and phosphoenol pyruvate kinase, no, not pyruvate kinase. Anyway, there's 2 of them, PK, P something, and Pepsi K. But those 2 enzymes counter the pyruvate kinase. That's the end of glycolysis, and then you have your fructose 1,6-bisphosphatase and your glucose 6-phosphatase, glucophosphatase, I believe is what it's called. All right. A futile cycle. That's if you're doing the same thing. So I put in glucose and I immediately burn it through glycolysis to make energy, and then I use that energy and go right back to make glucose. So that's a futile cycle. We're really not getting anything out of that. We're just kind of spinning our wheels futilely. So that's why we will see compartmentalization within our cells. Our glycolysis is typically cytosolic, and our gluconeogenesis, at least part of that, takes place in the mitochondria, and then back there so that we're not constantly spinning our wheels. Okay, so a lot of this is fairly straightforward. What I want you to know, I'm not going to go into any craziness in there. I'm not going to make you guys memorize the entire pathway. that comes later. You got to memorize one pathway for the semester. And I might actually do something different this semester and make that a quiz and do the exam a little bit different. Yeah. But let's talk about the take-home portion of the test. So the take-home portion of the test is going to be, very open-ended in there. Whereas you will need to have stuff at your fingertips for the in-class portion, I try to stick away from memorizing. It's like, you do not need to know that the delta G naught dagger of ATP hydrolysis is negative 34.1 kilojoules per mole. Nothing crazy like that. But the open books are not going to be find the answer somewhere. little asterisk that there later on there might be some find the answers, but they're not going to be in your textbooks. Yes, yeah. I want to see you use what's going on there too. And I'm a big fan of questions like, hey, so we know that pyruvate kinase, is regulated this way and catalyzes this reaction and is the final step in glycolysis. Suppose there is a mutation at this particular site. What do you think would happen? How would that affect glycolysis? Or maybe I'll be mean and say, how would that affect gluconeogenesis? You go, it won't affect it at all because it's not part of the gluconeogenic pathway. Ha ha, we're on to your tricks, Dr. Ebert. You have to write in that tone of voice too. I'll also probably pick one of those diseases that they cover in there, like maybe was it the glycogen storage diseases? There are something like 10 or 12 listed in there, and say, well, here we have a defect in this enzyme or this enzyme or this enzyme or this enzyme, and I might just pick one of those enzymes and say, actually, I got like 3 or 4 very ideas for questions for this. It's like, so suppose you have this mutation here that prevents this from binding something. Aside from putting in a brand new protein, do you think you could engineer a change that would facilitate binding or increase binding? How would this, or suppose you do something else? I'll ask you some crazy questions like that are very, very open-ended, and they don't necessarily have an answer. And eventually we're going to get to some questions that really just don't have an answer. I just want to see how you think and how you apply the stuff in there too. Like what happens if you go on the all bacon diet? It'll be delicious. Yeah, exactly. Yep. My son has a project in his for his middle school science where he's doing like it's not quite a Punnett square, but a predictive thing, and looking at family histories of medical things that affect different people going back a couple generations and the parents inside. And then there was a very interesting pool from that, because there are the people in my family who were obese, and then there were the people in the family who had high cholesterol, and they did not overlap. Well, there's like 1 or 2 people overlapped, but for the most part, the high cholesterol people were skinny and otherwise healthy, and the obese people, and then there's no heart disease either, and I don't think on either side of the family. So it's one of these weird things, and you kind of look at this and it's like, well, how do you explain this? Yes, but I look at like my dad's cholesterol is slightly elevated, but I mean I think he might be up to like 150 now, something. I don't know, he's just ballooning up in his 80s now. But he's always been very, very skinny. He's also always been very, very active. And it's been a long time looking at my grandmother's health as well too. Just eating horrible foods. It's like, oh, butter, a spoonful of butter. Not quite that bad now. I drink Mountain Dew because my dad drank a lot of Mountain Dew in there too. I had the idea of like going to the state fair and serving deep fried butter pats, but they actually do that now. You can get deep fried butter. I'm like, oh, damn it, there's my idea. I was going to be a cajillion. No, okay, I'd make like $50 selling deep fried butter. I don't think that's something you're going to make at home to eat. No. But anyway, that sparked a discussion of cholesterol. You get cholesterol from diet, you get cholesterol from your genetics, but you also manage cholesterol genetically as well too. And kind of like there is the ratio of ATP to AMP that signals the energy health level of the cell, there may be ratios of good and bad cholesterol. You talk about the LDL cholesterol versus the HDL, and it's one of these weird things where as I get older, I'm watching my cholesterol, and my LDL is my cholesterol sits there right at okay, this is high. It's right on the border. Sometimes it steps across, sometimes it walks back in there. It's one of those things in there. But my HDL, for the longest time was like 150% elevated. So the good cholesterol, super high. But good cholesterol is too much of a simplification. I think I'm actually changing. I have a short passage to read about that I've used in previous exams here too. But the ratio of those two may be important. And when we talk about the different components, the apolipoproteins and basically the lipoproteins in your body that deal with transporting cholesterol and sometimes fatty acids as well too, looking at the ratios of those as well. The very low density and low density lipoproteins tend to be the ones that will accumulate and will trigger macrophage attack and lead to plaque formation, whereas HDLs are more involved in clearing excess cholesterol. So it may be a ratio. It's like, yeah, eat all the cholesterol you want as long as you can clear it as fast. And that just may be some crazy weird genetic component that apparently on my father's side extends back at least a couple generations, looking at people there too. So I also wonder, because this was a habit I did not pick up. I picked up Mountain Dew, but my dad was a smoker, and I don't know, he might still, yeah. But I mean, phenomenally healthy. I've known other people who, it's like people who will develop COPD or lung cancer is a horrible thing there as well too, and it's just he's fine. For working on a farm and smoking a whole lot, and then working in a paper mill and air quality and all sorts of stuff like that. It's apparently he's got phenomenal lungs. He used to smoke Winston's, I believe.
Oh, that's why.
Yeah.
That's why, because people that smoke Newport, like, so my grandmother, actually my great grandmother, she's 78, she never had any of my smoke cigarettes as long as I've been alive. But she smokes a certain, it's like a very hard brand of cigarettes. I call them guilty cigarettes because like she never had any issues. Like it's so weird.
There's probably a huge genetic component to that. Because it's crazy. Although I have to comment, Virginia Slims, smoked by little old ladies everywhere, because one of the ingredients, and I don't know if this is still true, but one of the things that they used to help cure the tobacco was like basically an elephant tranquilizer. So I don't know if they still include some of these compounds in there, but there are so many things that are added to tobacco during the preparation process. But yeah. I wanted to say it was ketamine, but I don't think it was ketamine that was in there. That's a little too crazy. There was some weird, unusual compound, but they used it as an elephant tranquilizer at zoos or in Africa when they needed anesthetized. So that was one of my friends commented. That's why the little old ladies who smoke Virginia Slims are always so calm. Yeah. But anyway, so like I say, the exam itself, the in-class portion is kind of short. I like having an hour and a half because I could ask way more questions than I do. But the take home, like I say, we'll plan for next Tuesday. You'll have the in-class portion, and then the take home will be due back on Thursday. And I hope to replicate that. Tuesday will be exam day, and Thursday will be turned back in. The take home is usually not too awful. I try, and I could take home. I can give you 20 questions, right? Oh, I just murder any love you have for biochem. But it's usually somewhere around 10-ish, 8 to 12, something like that. And then a lot of times I will give choices to like pick 2 of the following 3 questions, or I think there's one section where I'm like, pick 2 of the following 5 questions. So they're just crazy. So I try not to make the exams awful, but that does not mean that they are fun, because there's just so much information here. And the biggest challenge that I think students in biochemistry will have is figuring out, how do you take this massive amount of information and organizing it so that you can understand it? Yeah. And it's tough. It's
It's so true. Like, where do I put all these?
Yeah.
My brain is only so big.
Yeah. I had a joke. If I could get rid of all like the 80s rap lyrics that are still stuck in my brain. Yeah. So that's it. You need to come up with exactly. Yeah. That's right.
I don't know.
All right.
I use I it's a word for it. I can always forget the word. Like I've set these reminders in my head, like
mnemonics.
You see this.
Yep.
So like some years ago, I'm like, when you get home, if you see the door, do this. So when I get home, okay, so that's how I.
Yep.
How I remember things.
mnemonics, and like the old thing was like tying strings around your finger to remember something as well too. But yeah, yeah. My one of my friends is trying to train his dog to respond to different tones. And anytime when he hears this tone, it's time to come inside, something like that. So yeah, you can do that. You can play different sounds on your phone at certain times too. Oh, okay, that reminds me of this. But, yeah. Yeah. Check this real quick. Oh, okay, good. All right. So let me look at one other thing here real quick. Let's see if this will help you. Your conversion of what's the name of it? Okay. Okay. I think I've yeah, almost certainly. I think I've heard of something. I know there's something that's supposed to promote that. And and so this is where we'll go. We're going to talk about this later on as well too. But yeah, Lance Armstrong, the great American hero, 8 Tour de France's, doping, just like everybody else in the Tour de France. But yeah. I mean, I still remember there was one year, and it was always fun for me to watch the. I have no interest in cycling, and the cyclists I've met are just not pleasant people for the most part either. There's a handful of exceptions, but most cyclists, like you're just from Jersey or something. No, sorry, I met a lot of people from Jersey who are not very nice either. So that's my stereotype of Jersey. I used to joke that I gotta find somebody from New Jersey who's nice, and I finally did. I'm like, okay, it's not just full of terrible people. Now, yeah. Now my quest is to find a nice guy from Poland, because every guy I've met from Poland has been a flaming jerk. Just yeah. And it's funny because I've met women from Poland who are nice, but the guys from Poland, yeah. There's a lot of it. But anyway, a lot of these, like the whole doping thing, we can talk about that in here. It's like how to dope efficiently. That's what you can learn from biochem. I mean, go into sports medicine. Yeah. But I think one of the things that maybe helped Lance Armstrong was when he went through the cancer diagnosis and the treatment for that. And then you can ramp up, it's like, oh, all these cells were taken away. You can rebuild them back even better. And then erythropoietin was a big thing for several years in there because, monitoring erythropoietin, you're basically the red blood cells and how fast can you get lactic acid from the muscles back to the liver and regenerate it in there, so you don't get that burn, so that you can keep pedaling more and more and longer and longer and all that. And yeah. Yep. And and there's all sorts of like training methods. I think there was like one guy from somewhere in South America who did all his bike training in the Andes or something. So it was a high level, so that he was used to consuming and using oxygen, and that helped. But the thing is, apparently, the whole make the Russian Olympic Federation look clean now. But yeah, that's the thing. Everybody wants a competitive edge. I'm sure here in a few years, well, see, these are mostly adults, or at least adolescents moving into adults who are making decisions to do stuff to. You think of like steroids from 20, 30 years ago as all the rage, and then these EPO alterations and supplements and doing stuff like that became a thing. And there's a constant race between catching what people are doing and then finding new ways to enhance performance. So why not do a little CRISPR-Cas9 and increase this gene production, or turn on stuff like that. And then you get into an area of theoretically you could have a responsive thing where like what's that blue light. I always think of like the dentist where, they put the enamel on there and then they put in the certain frequency light and that hardens, the sealants to protect the tooth. So you could theoretically do something under the skin like that as well too, where, maybe in helmet would probably not be useful, but you could actually have something there where it turns on a certain frequency of light to stimulate something to release, to basically, upregulate a lot of these enzymes that are involved in athletic endeavor. And is that cheating? Well, it's not natural, but then if you make it genetic, and just because you can turn it on and off, and who knows? So maybe that's where I should go. Yes, yeah, and that's what, and with Lance Armstrong, and just the few samples that came back as questionable were litigated and challenged and so on and so forth for such a long time. And it's like, I was like, yeah, boy, geez, it certainly seems like the French have it out for him, But then it turns out, oh, okay, and then the few people who, I actually read some stuff about him and how one of the people who just has a wonderful reputation, blanking on his name now, but actually it might have been Greg LeMond. It just kind of like got pushed aside and threatened because he wouldn't go along with stuff that was in there. But I had to mention, this will be my tie up here. I had the best Halloween costume a couple of years ago when we still actually had Halloween. Took my daughter to Old Salem in her costume, but I dressed up. And it's like I have a bike and I ride. I don't have any like the fancy stuff in there, but I tried to find something that was a bike uniform and a big helmet and the little glasses on there. That was the big thing there too. So I wore that, and I also had a picture of like various rectangles of different colors, and I hung that around me while wearing the biking apparel. And then I would ask people, "Who do you think I am? What costume am I?" And it's so freaking obscure because you got to go back to the 80s was when Greg LeMond was famous. He was like the first American to win the Tour de France, and won it like twice. So Greg LeMond, awesome. And there's also an artist called Mondrian, and his big thing was painting these different colored squares, kind of like Rothko or one of these newer artists. I was Greg LeMondrian. And the best part was one person got it. A couple people were making the connection and got in there too. It was just so fun. So it's fun to be obscure, I guess. That's why I have a PhD in biochemistry, so I can make puns about stuff. It's like my Cobra Kai joke. That's even more obscure.